Imagine discovering that the seeds of a deadly cancer were sown decades before any symptoms appeared. That's the startling revelation from a groundbreaking study published in Nature Genetics, which has mapped the DNA damage timeline in multiple myeloma, a relentless blood cancer and the second most common hematologic malignancy globally. This research doesn't just chart the disease's progression. It rewrites our understanding of how and when it begins, opening doors to early detection and personalized treatment strategies. But here's where it gets even more fascinating. The study reveals that the genetic mutations driving this cancer can start accumulating a staggering 20 to 40 years before diagnosis. This extended preclinical phase isn't just a footnote. It's a potential game changer for intervention and prevention. Multiple myeloma is notorious for its complexity, often lurking silently for years in a phase called monoclonal gammopathy of undetermined significance, MGUS, before progressing to smoldering myeloma and, finally, symptomatic disease. Until now, unraveling the precise sequence and timing of the genetic changes driving this progression has been like solving a puzzle with missing pieces. This latest research, however, sheds light on how DNA damage accumulates over decades, offering a chronological map of the disease's evolution. And this is the part most people miss. By understanding when these mutations occur, we can potentially intercept the disease long before it becomes life-threatening. At the heart of this study is a treasure trove of data, 421 whole genome sequences from 382 multiple myeloma patients, primarily newly diagnosed, but also including some longitudinal samples post-treatment. To reconstruct the timeline of mutational events, researchers employed a cutting-edge computational tool called the molecular time model. This model acts like a genomic stopwatch, measuring the accumulation of benign point mutations, harmless changes that occur at a predictable rate and serve as timestamps in the tumor's history. By analyzing these mutations, scientists can pinpoint when critical genetic alterations occurred, effectively mapping the tumor's evolutionary journey. Here's how it works. DNA accumulates point mutations at a relatively steady pace over time. When a chromosome duplicates, a hallmark of multiple myeloma, it carries with it a baseline of these benign mutations. Over the years, each duplicate copy accumulates its own unique mutations. By comparing the mutation burdens in these copies, researchers can estimate when specific chromosomal duplications or rearrangements took place, creating a detailed timeline of tumor evolution. This approach not only confirms existing theories but also refines our understanding of key events like hyperdiploidy, the gain of multiple chromosomes, which is now clearly linked to an earlier translocation involving the immunoglobulin heavy chain, IgH, locus. This IgH translocation emerges as a pivotal starting point in about 10% of cases, shaping the disease's trajectory from the outset. But here's where it gets controversial. The study highlights the role of treatment itself in shaping the genomic landscape. For instance, exposure to melphalan, a common chemotherapy drug, appears to accelerate the acquisition of specific mutations, such as the gain of the long arm of chromosome 1, CHR1Q gain. Patients who develop this alteration early in their disease course face significantly worse outcomes compared to those who acquire it later. This raises a provocative question. Are we inadvertently driving genomic changes with our treatments, and could this be complicating the disease's evolution? The implications of this research are profound. It underscores the inherent heterogeneity of multiple myeloma, not just biologically but temporally, emphasizing the need to integrate timing into clinical decision-making. By knowing not just which mutations occur, but when they occur, clinicians could one day tailor therapies to target vulnerabilities unique to each phase of tumor evolution. Imagine predicting treatment resistance by tracking mutational dynamics over time, or estimating patient survival with unprecedented accuracy based on mutational chronology. This isn't science fiction. It's the future of precision oncology. Yet the study also raises fundamental questions. How do early DNA damage events influence later mutations? Are there other genomic markers with similarly impactful temporal characteristics waiting to be discovered? Could intervening during the prolonged latent phase alter the disease's course or even prevent progression? These questions set the stage for a new era of research, one driven by temporal genomics. This breakthrough was made possible by a multi-institutional collaboration among leaders in computational biology and genomics, including the Sylvester Comprehensive Cancer Center at the University of Miami, Memorial Sloan Kettering Cancer Center, and the German Cancer Research Center. Their integration of large-scale whole genome sequencing with advanced molecular modeling showcases the power of interdisciplinary research in deciphering cancer's complexities. In essence, the work by Kadura, Landgren, Diamond, and their colleagues represents a monumental leap in understanding multiple myeloma's evolutionary timeline. It reminds us that the tumor's genomic identity is shaped not just by the events themselves, but by their sequence and timing, a dimension often overlooked, but with profound therapeutic implications. As precision medicine advances, incorporating the when alongside the what in genetic alterations could revolutionize patient care. For those eager to stay updated on this evolving field, the Inventum blog and Sylvester Comprehensive Cancer Center's social media channels offer ongoing insights. This narrative of temporal genomics brings hope that future myeloma therapies will be more personalized, effective, and timely, ultimately transforming outcomes for patients battling this challenging disease. But what do you think? Does this research change how we should approach cancer treatment? Could early intervention during the latent phase be the key to preventing progression? Share your thoughts in the comments. Let's spark a conversation that could shape the future of oncology.